Hey everyone, my name is Danny Lee and today we'll learn about genomes. Genome? Nah, I'm talking G-E-N-O-M-E, genome. But in order to understand what genome is, you need to first understand what a DNA is. As you all know, a DNA molecule is composed of sugar phosphate backbone, 4 different nitrogen bases, guanine, cytosine, adenine, and thymine. And in between them there are hydrogen bonds. Guanine and cytosine always act as a pair, and same thing for adenine and thymine. In normal state, DNA is tightly packed into a structure called chromosomes. Now in these chromosomes there are certain parts called genes. Genes are segments of DNA. They contain instructions for our individual characteristics from our parents, like hair color or skin color. Okay, do you remember the 4 nitrogen bases? They're like materials from a Lego instruction manual. The order of these bases tell us how and when to build certain proteins. So after all this, what is a genome? Genome is an organism's complete set of genetic instructions, so all of the genes. Each genome contains all the information needed to build that organism and allow it to grow and develop. Let me give you an analogy. So this book called Genome has 23 chapters representing a chromosome. In total it has 3 billion letters, the nitrogen base pairs. There are many copies of this book and each of these books are most of our cells. In 2003, the Human Genome Project was completed, meaning they figured out the orders of all the bases and all the genes. But how? I'm about to show you. This is called genome sequencing. First, you sample DNA, cut it into fragments using a restriction enzyme. Just one copy of DNA fragment is easy to lose, so we use a plasmid DNA of a bacteria to replicate it rapidly. Dump all that in a centrifuge, which is sort of a blender. At this point, the DNA can be separated from everything. DNA fragments are double-stranded at first, but we need them to be single-stranded. So we heat them up, breaking the hydrogen bonds. DNA primer and DNA polymerase are added. Polymerase staples nucleotides to the primer. Normal nucleotides keep attaching and the strands gets longer and longer. But once in every while the dideoxynucleotide comes along. This bad boy, also known as the terminator base, has a structure designed to stop nucleotides attached to it. Once this attaches, polymerase just falls out. Temperature increases again, the strand just falls out. Repeat this process over and over and over. Because the position where the terminator base attaches is completely random, if you repeat this process enough, it results in all different lengths of strands with different terminator bases at the end. Each terminator base is labeled with a color, which is detected by a laser. These strands travel through a charged capillary. Shorter ones travel faster because they're lighter. At the end, because they put it to shortest to longest, we now know the order of every base in that DNA. Genome is like a puzzle of life. The pieces of puzzle come together to tell a story about human beings. That fascinates me, and I hope it fascinated you too. My name is Danny Lee, and thanks for watching. Genome.